Let's continue learning by talking more about inheritance. A nucleus that has 2 pairs of chromosomes, one from each parent, is considered diploid. In humans, that number is 46, or 23 pairs. We get 23 from our mother and 23 from our father. We find the diploid cells in somatic or body cells. The gametes or sex cells have only 23 chromosomes, and that is called haploid. They are called haploid because the chromosomes do not come in pairs, but rather have a single chromosome that derives a pair of chromosomes. We use the variable n to represent the haploid number and 2n to represent the diploid number. For humans, each egg and sperm are haploid, n equals 23, so that when the egg is fertilized with sperm, the resulting zygote has a total of 46 chromosomes. This pattern of inheritance from diploid to haploid and back to diploid is seen in all organisms that undergo sexual reproduction. These organisms, like humans, are all derived from eukaryotic cells. Gregor Mendel was an Austrian monk and worked with pea plants in the mid to late 1800s. In 1865, he published results of his experiment with pea plants and how their characteristics were passed on to offspring. At the time that Mendel was working, the term gene was unknown, as was DNA and the structure of DNA. Mendel used artificial pollination to obtain sperm cells in the pollen of pea plants to fertilize the egg cells of another pea plant. He examined 7 different characteristics, as is shown in the table. He consistently found nearly a 3-to-1 ratio of one plant to another. For example, looking at the seed shape, he found the ratio of round to wrinkled as 2.96 to 1, which we can round up to 3 to 1. Let's look at another characteristic, plant height. Mendel took purebred tall plants and crossed them with purebred short plants. Purebred means that tall plants only had the characteristics of tall plants, and short plants only had the characteristics of short, as did the parent plants of each group. He wanted to see if he got all tall plants, all short plants, or a mix of short and tall when he crossed the purebred tall with the purebred short. The result of this cross were that all of the offspring were tall. We know now that in pea plants, the allele, or the version of the gene, for tall plants is dominant over the allele for short plants. A purebred or homozygous plant has the same alleles. For example, a purebred tall plant would have uppercase T, and a purebred short plant would have lowercase t. A plant that has both is called heterozygous and would show uppercase T, lowercase t. To show these crosses, a Punnett grid or square can be used as a tool. You can see an example here. The purpose of a Punnett square is to show all the possible combinations of genetic information for offspring of a particular trait. If only one trait is examined, that is a monohybrid cross, while 2 traits would be dihybrid. There are a series of steps when constructing a Punnett grid. 1. Choose a letter to represent the alleles. It should be the same letter for both. The dominant trait is represented by the capital letter and the recessive by the lowercase letter. Choose letters that make an obvious distinction between the uppercase and lowercase letters. For example, the letter B works well, but not the letter O. Number 2. Determine the parent's genotype. There are 3 possibilities: homozygous dominant, uppercase T, homozygous recessive, lowercase t, or heterozygous, uppercase T, lowercase t. It is very easy to determine the genotype of the short plant, lowercase t, as that is the only way the trait of the short will be expressed. It becomes more difficult with the dominant trait of tall, as there could be 2 genotypes, uppercase T, homozygous, or uppercase T, lowercase t, heterozygous. Both will be expressed as tall. The parental generation, or P generation, is what is being crossed. The offspring produced are called the first filial generation, or F1. Crossing 2 members of the F1 generation will produce the second filial generation, or F2. Number 3. The third step is to determine what gametes the parents could produce. If the parent is uppercase T, the only gametes that can be produced are those with the uppercase T, or the dominant trait. The same occurs when the parent is homozygous recessive, lowercase t. Only egg or sperm with a lowercase t can be produced. If the parent is heterozygous, uppercase T, lowercase t, they can produce 2 different gametes, one with an uppercase T and one with a lowercase t. Number 4. After the previous steps have occurred, you can draw the Punnett grid, or the Punnett square. The parent's gametes are placed on top and the left side of the square. Next, you fill in the squares by pulling in one allele from the top and one from the side to place 2 total alleles in each box. Each square represents one of 2 possible outcomes. You can see in the cross between 2 parents that are heterozygous for height, there is a 25% chance, or 1 in 4, of being homozygous dominant, which is tall, uppercase T, uppercase T. A 25% chance, 1 in 4, of being homozygous recessive, which is short, lowercase t, lowercase t. And 50%, 1 in 2, chance of being heterozygous, uppercase T, lowercase t, which is also tall. It is important to remember that this shows probability. It does not mean that if one plant is short, the others have to be tall. When we look at plants specifically, it's important to understand their anatomy, as it shows how each plant can sexually reproduce. Flowering plants, like pea plants, produce pollen, which carries the male genetic material. In addition to that, flowers contain structures called ovaries that house the female genetic material. The transfer of the genetic material from the pollen into the ovary will result in sexual reproduction and create a new seed that can then grow into a new plant of the next generation. The process of pollen moving from one flower petal to another usually requires another organism, like an insect, to be a pollinator. Take a bee, for example. It moves from flower to flower, taking the nectar, and also collecting some of the pollen from one flower and dropping it off to the next. This can support the cross-pollination of one plant to another plant, and can also support self-pollination if pollen from a plant enters the ovary of the same plant. Self-pollination usually results in less genetic diversity compared to cross-pollination. It is important to understand the difference between alleles, genotype, phenotype, homozygous, and heterozygous, some of which we have already mentioned on the previous slides. An allele is a specific form of a gene and differs from other forms of the same gene by a few base pairs. For example, there is a condition where a person is unable to see any color. This is not color blindness, which is a sex-linked disorder discussed later in the video. The condition is called achromatopsia, and people with it cannot see color because they are unable to produce the protein transducin in their retinas. You can see an example here based on these 2 renders of the same image. This is caused by a single base pair difference between the most common allele, with a C at position 235, and the rare mutation of a T at position 235. This shows 2 different versions or alleles of that gene. A genotype is a symbolic representation of a pair of alleles, usually using letters. For example, uppercase T, lowercase t, or uppercase T, lowercase t. Genotypes are always expressed with 2 of the same letters that represent the same gene and the alleles that make up the genetic code for that gene. Homozygous means you have 2 identical alleles, either both uppercase or both lowercase. Uppercase T, T, is homozygous dominant, while lowercase t is homozygous recessive. Heterozygous refers to having 2 different alleles, uppercase T, lowercase t. A carrier is an individual who has a recessive allele, but it does not have an effect on the phenotype, which we will continue to discuss throughout the video. The phenotype is the observable trait of an organism based on the genotype. In other words, the phenotype is what you see. For example, in a plant with a genotype of uppercase T, or uppercase T, lowercase t, you will see the phenotype of tall plants, because there is at least one dominant allele present in each genotype. If the genotype is lowercase t, the phenotype is a short plant because it only has the recessive short alleles. There are some phenotypes that can change due to environmental factors and others that are controlled by genetics alone. Phenotypes controlled by genetics alone include your ABO blood type. Environmental factors cannot change that. An example of a phenotype produced only by environmental factors may be learned behavior, such as birds learning new songs. An example of a phenotype that may be due to a combination of genetics and environment is height. A person may not reach their maximum height if they do not have the proper nourishment obtained from their environment. We have used the term dominant and recessive a few times so far in this video. Let us go a little deeper into what these terms mean. A dominant allele is an allele that has the same effect on the phenotype, whether it is with the same allele or a different allele. If the dominant allele is present, it is what is expressed. In our example of pea plants and height, if the offspring have 2 alleles that are both the dominant allele, uppercase T, the phenotype is tall, which is the expression of the dominant allele. If the plant has 2 different alleles, with one being dominant and one being recessive, uppercase T, lowercase t, the phenotype is tall as well, which is again expression of the dominant allele. The recessive allele only has an effect on the phenotype when there are two recessive alleles and no dominant allele present, lowercase t, lowercase t. This sums up what the concept of dominant is all about. Dominant alleles are always expressed when they are present, and the recessive alleles are only expressed if there is not a dominant allele present, which would mask the recessive allele every time. Sometimes organisms can express their phenotypes differently depending on the environmental factors present. This is called phenotypic plasticity. As an example, some birds can activate genes to produce more maltase when their diet has more grains and less insects, while other birds can modify their foraging behaviors depending on the types of food available. In either case, changes in the environment are directly impacting the genes that are silenced or expressed in the organism impacting phenotypes. Phenology is a term used to describe cyclical events such as food availability, which can provoke these changes within organisms. Phenotypic plasticity can generate changes in several ways, through physiology, such as the enzyme example, and behavior, such as foraging behavior. Another way is morphology. One example is the freshwater snail, Physa virgata. The snail normally has an elongated shell, but when its predator, the bluegill fish, is present, a phenotypic change occurs. The genes are expressed slightly different, causing the shell to be less conical and more rounded and shorter. This makes it more difficult for the bluegill to crush. This is not considered a mutation, as the genotype does not change. The snail still has the same alleles, but it is able to silence and express genes in different combinations to change the shape of the shell. There are many recessive genetic conditions which affect organisms. In humans, one disorder is phenylketonuria, or PKU. A recessive disorder can occur even when both parents do not have the disorder, but they must be heterozygous for the disorder, meaning they must be carriers. PKU is caused by a mutation in the autosomal PAH gene, which results in low levels of the enzyme phenylalanine hydroxylase. This enzyme is necessary to convert the amino acid phenylalanine into tyrosine. Phenylalanine is toxic and can build up in the brain and impair development of the brain, which is why converting it into tyrosine via the enzyme is so important. Individuals who suffer from PKU do not create this enzyme and therefore have no way to break down phenylalanine. When diagnosed at birth, modification in diet can prevent the impairment. Children affected by PKU must have diets that limit phenylalanine and will need supplements to be sure their diets have enough protein. Looking at the Punnett grid here, you can see that both parents must be heterozygous for the mutation, and while they do not have PKU disorder themselves, they are carriers. There is a 25% chance that their offspring will inherit both recessive alleles and have PKU. These disorders are called autosomal recessive disorders because they are caused by recessive alleles. Autosomal means that the disorder is found on the first 22 pairs of chromosomes and not the sex chromosomes. Other examples of autosomal recessive disorders in humans include albinism, cystic fibrosis, sickle cell disease, Tay-Sachs disease, and thalassemia, all of which are found on different chromosomes and have different impacts on the body. A gene pool is all the genes available to a breeding population, and there are any number of alleles of a gene that can be present in the gene pool, but an individual only inherits 2 of those alleles. We have discussed only 2 possibilities, either dominant or recessive, but genetics is not always so simple. Sometimes there are 3 or more alleles for a trait, such as in blood type. This will be discussed on the next slide. Our immune system has genes called HLA genes, and some of them have dozens or even thousands of different alleles. This is beneficial to use when you think of all the different pathogens our bodies need to fight. Single nucleotide polymorphisms, or SNPs, called SNPs, occur when a nucleotide of the genetic code, like C, is not found where expected, and at that location is instead a different nucleotide. If this change is found in part of our DNA that is a coding section, it could be that a different amino acid is coded for, which could change the structure and function of the protein that is translated. One example of this in humans is the ability to taste bitterness. This is important to survival as many toxins are bitter, and the ability to taste this will protect the individual from consuming the toxic substance. The molecule, PTC, or phenylthiocarbamide, is a bitter substance. People who have one version of the gene, TAS2R38, can taste the substance. This gene encodes for a protein, taste receptor 2, member 38, which allows a signal sent to the brain for interpretation. In order to taste PTC, only one functioning gene has to be present. Because there are several different SNPs of this gene, there are multiple phenotypic outcomes, which include some people being able to fully taste, some people can intermittently taste, and others who cannot taste it at all. It is common for biology classrooms to have their students try PTC paper to see what type of phenotype they possess. The ABO system of blood typing is an example of multiple alleles. Be careful here, as there is still only one gene that codes for blood type, but there are more than 2 alleles possible. While the blood type system in humans has 3 alleles, there are 6 possible genotypes, with 4 possible phenotypes. While blood type demonstrates multiple alleles, it also demonstrates codominance. We use the letter I, uppercase and lowercase, to represent the multiple alleles, and we attach superscripts to distinguish between them. Uppercase I, superscript uppercase A, uppercase I, superscript uppercase B, and lowercase I. Uppercase I, superscript A is used to represent the allele that produces a protein called type A antigen, which gives the person type A blood. Uppercase I, superscript B is used to represent the allele that produces a protein called type B antigen, which gives the person type B blood. And lowercase I is the recessive allele that produces neither A nor B antigen, giving the person type O blood. A person with 2 uppercase I's and superscript A's, or 1 uppercase I, superscript A, and lowercase I, will give the phenotype A blood. A person with 2 uppercase I's, 2 superscript B's, or 1 uppercase I, superscript B, and lowercase I will give the phenotype B blood. 2 lowercase i's will give the phenotype O blood. When the antigen for A and B are both present, codominance will be evident and a new phenotype will appear, type AB blood. You will notice that the antigen on each cell is always opposite the type of antibody that is in the bloodstream. This is because when an A antigen and an A antibody get together, they end up connecting and clotting, which is a protective measure built into the system in case any foreign blood enters your body. For this reason, it is important that if you were to receive blood from a donor, you need to receive it from a blood type that will not clot with the antibodies present in your bloodstream. This makes some blood types great for donating, like type O blood. Because there are no antigens present on the blood cells, there is no way for it to clot with any antibodies, making someone who has type O blood a universal donor.